Hi again, everyone. After Wednesday talks today, talking about database, and after Joanne's great introduction, I think this will work perfectly. So let's get in, and I will talk really specifically about IMGVR. First, the framework, more general, like the IMG framework, I should say, within which IMGVR works. Then IMGVR per se, data and features, and finally something about the current interface and how you can use it. To understand where IMGVR comes from, I need to understand a little bit, or I need to explain a little bit what JGI is. So we are a DOE Office of Science user facility, which means we are here. Part of our mission is to, like it's written here, enabling researchers to solve like energy and environmental challenges, which is one of the reasons why we don't work a lot with, for instance, human gut data. We are really focused on microbial ecology / energy and environment. And so the idea is, as a user facility, we are here to give access to some of the most advanced capabilities, and our area of expertise, especially, is integrative genome science. And you have different calls for user projects. So users and pretty much every one of you here could get access to our capabilities. If you are interested into this, I won't go into any details, but like shoot me an email, DM me on Twitter or whatever, just like reach out, and I will be more than happy to kind of check with you if we could work with you, basically. So what do we mean by integrative genome science capabilities? We mean a lot of different things, but specifically for my purposes, we mean microbial genome sequencing, especially microbial metagenome. And every time I will use metagenome in this talk, I think, or I mean metagenome as a broad sense, meaning metatranscriptomes. I mean pretty much any multi-omics, basically, which is done at the community level. And so when we started doing genome sequencing and metagenome sequencing, one of the questions that popped up was, how do we best share and release this data, and how do we optimize and analyze, both analysis and the annotation of our own data that we keep generating? And so that's the origin of IMG, which is really the data management and analysis platform for microbial genome that JGI developed to basically handle our own data that we kept producing. So this article kind of tells you that it started in 2006, so that has been a long time. The current IMG, now called IMGM, because it's now integrated microbial genomes and microbiomes to kind of, expand to metagenomes and similar types of data sets, is now in its version 6. This, if you are curious about the database itself and the interface, like the whole of IMG is described in this NIR database special issues. And if you have if you want a sense of scale, this is not IMGVR, right? This is like the whole of IMG, but these numbers are just staggering. I won't even try to read them, but like we have billions basically of genes, scaffold, bases, whatever. So what do we have in IMG? And that's one of the important part I need to explain so that you can understand where IMGVR data comes from. IMG hosts, in terms of metagenome, 3 types of contigs. We have all. Then we have what we call like relevant metagenomes that we import from other databases. And I mean relevant here back to our mission, right? Like energy and environment. So, we tend to import from other databases like NCBI and EBI. We tend to import metagenomes linked to this, different environments, surveys, and things like this. And finally, we do have in IMG a number of user-submitted metagenomes, because as an IMG user, you can submit your own data, analyze it, and then eventually release it publicly, and in which case it kind of goes into this big pile of IMG public data. So that's all to say we have a lot of publicly available metagenomes on IMG, the bulk of it coming from our own sequencing of user projects. Now, where do IMG comes from in all this? Of course, when you have so many metagenome assemblies and metagenome contigs, one of the questions that at one point comes on is, do we have any cool viruses in there? And so that's really what triggered this large-scale mining of viral contigs in all the publicly available metagenome at the time, which was 2016. And that's the Earth Virome paper that Joanne already mentioned in 2016, led by David Paeth. And he was using a custom detection approach. That's for people who are already here on Wednesday. We talked about this very, very briefly, but like this Earth Virome protocol is actually described in this Nature protocol paper if you are curious, which primarily relies on marker genes, which are, these VPFs that you also heard about yesterday, if you follow Joanne talks. I shouldn't say we actually. David's project recovered more than, about 125,000 viral contigs, which was a lot, to analyze and everything. But then came the question of how do we share this resource in a meaningful way beyond, dumping everything in a supplementary file with this 2016 papers and moving on. So to share this, more than 100,000 contigs, it was decided to create a subset of IMG and call it IMGVR. Basically, IMGVR is part of the big IMG family, but it's like a separate portal in a way that gets you access only these contigs that we deemed were most likely to be viral. First iteration came in January 2017 with an updated input dataset from this 125,000 a little bit. Then June 2018, we released the version 2. Again, updated input dataset. What I mean here by updated input dataset means we had more public metagenomes that had come in, so we mined them and we added all the contigs to the pile. In 2018, we also introduced first kind of very custom, but like the beginning of an approach for quality estimation, meaning trying to distinguish what is a partial genome versus what is a complete or near complete genome. And it also started to integrate better into the whole IMG ecosystem. And nowadays, and what I will describe today, is now the third iteration of the IMGVR database, which was released in about a year ago, September 2020, which we call IMGVR v3. Okay, a quick sense of scale. I like to show this basically because it's a pretty easy graph to understand. The blue line is all the virus genomes, in this case from NCBI, basically. And the yellow line is the growth of all the viral contigs extracted from metagenomes. So it can be partial or near complete genomes. At this point, I'm just counting, not even non-redundant, I'm counting all contigs. But still, it gives you a sense for like, with the release of IMGVR1, then 2, then 3, how quickly we accumulate viral genome information when we mine metagenomes and viromes and all of this metagenomic data. And I can't share numbers yet, but the next iteration of IMGVR will roughly be on the same trend, basically, of this exponential growth. Okay, so that's the context in which IMGVR exists and was created. What does this host right now? What is really the content and the meat of IMGVR v3? Like I said, a majority of the data comes from these IMG public metagenomes, and a lot of them comes from this viral contig detected with this Hertz virome protocol. At this point, we have not yet switched to another virus detection tool for a number of reasons that I'm happy to discuss later if anyone is curious. But that's, for you to know, if you look at all the IMGVR data represented as these boxes on this right panel here, a lot of them, 1.9 millions of the 2.3 millions comes from this viral contig detected based on this Hertz virome protocol from IMG public metagenomes. Then we try to add to this some relevant data set. Like Joanne was saying, you're trying to add to your database as many sequences that could be useful as you can. So I will just go quickly through this. The first of this kind of extra data set is a prophage data set we published in 2014, where we had mined gene bank data at this time for prophages using VirSorter1. The second one is a prophage of a database of innoviruses that we released in 2018. We built this database by mining public IMG genomes and metagenomes, but because innoviruses, which are filamentous bacteriophages, are not well recovered by pretty much all of the standard virus detection approaches, we had our custom approach. A bit of the same for giant viruses. We added to IMGVR the database of about 2,000 giant virus bins, which had been mined, built, and analyzed by Frederick Schultz in 2020. A bit of the same thing. We know they are not well recovered by most of the standard pipelines, so we also know they were in this data, so we wanted to add them. Same story here. We had this virophage custom detection pipeline that we developed in 2018, and we already had this data. We knew they were in IMG, so we also made sure that they were no part of the IMGVR. And then finally, for anyone who has kind of looked deeply into the CheckV manuscript, which I will come back to CheckV as a tool, but it does use a custom database of predicted complete genomes, meaning what I tend to call circular contigs, which Stephen is right, should be called contigs with direct terminal repeats. It's the same thing, basically a contig with identical ends, which signs somewhat like, usually signs a complete genome being assembled in one piece. And these complete genomes are very important for CheckV as a tool. And so Stephen went through the process of finding as many as he could from public metagenomes in IMG and other databases. And for this, to detect which of these were viral, he again used somewhat of a custom approach. So the conclusion of all this is, one of the unfortunate, but it is what it is, aspect of IMGVR is a lot of different detection tools have been used, and not all sequences have been detecting using the exact same cutoff or thresholds. And that's the price we pay for trying to be really exhaustive and as comprehensive as we can in a way. Now, added to that, you have sequences that are not in IMG, so that are not visible online when you use the web interface, but we still included them into the database as references for when we do, host prediction or VOTU clustering, because there are some either very important information like, isolate genomes from RefSeq and GenBank, or because they are, well-curated and important data sets of UVic from metagenomes, except these metagenomes are not on IMG. So these sequences are mostly like in this right part here. In this published UVics, not in IMG, it's mostly the global ocean virome 2, the gut virome database, the uncultivated gut virus, which are like a bunch of like collection of viral contigs from human gut metagenomes that have been published recently. And then for all of these intermediary data sets, you always have a mix of like IMG sequences and sequences not in IMG. So that's another thing that you have to understand when working with IMGVR, is there is a slight discrepancy between what you can see online and what you get in the downloadable database. What you can see online is everything that is on IMG as a contig. What you can download is the same, plus this extra that we used for reference purposes, but cannot be shown on the web page because the sequences themselves are not in the IMG system. Okay, 2.3 million sequences, awesome, job done, right? Of course not, and of course no one wants to work with the 2.3 million sequences every time. And the thing is, not every one of these 2.3 million sequences is useful, or at least depending on your purpose, you may want to filter them down. So what we have tried to do with IMGVR v3 is giving you some information on each sequence to help you filter out and basically select a subset of the database, exactly like Johan was describing. So the first step, and I will just go through like briefly what analysis, quote unquote, pipeline we use for IMGVR. First step is post-processing. We remove contamination, and we try to estimate completeness of the genomes using CheckV. So for this, we especially rely on this MyUVic paper where we describe what would be like a good criteria for what is a high quality genomes versus low quality genomes. And, I will let you read through this if you are curious, but it's basically, all about completeness of the genome. Do I have a near complete or complete genomes versus do I have a small fragment of a genome? Just a quick data point. Once you do this, you can check in your IMGVR v3 how many sequences are high quality versus low quality. And as you could expect, the high quality genomes and near complete genomes are only a small subset of IMGVR, but still, yes, it's only 10%, but it's 10% of. So user can now identify the 10% high quality, and 10% of such a big number of contigs is still a lot of contigs. So that's kind of how we see things. Is like, by providing you this information, you as a user can go in and select if you want to work only with the high quality or only with the one that are more than X% complete, rather than tackling the whole 2.3 millions. Then we cluster into VOTUs, again using CheckV based on pairwise global ANI. Another piece of data here, we have done like some quick kind of accumulation curves or rarefaction curves, depending on how you want to call them, using all of IMGVR, V2, and V3. And as we accumulate more viral contigs, basically, do we see less and less VOTUs? And the results is not really like we have no plateau inside, and it's kind of matches what Johan was just saying about like even our example with soils. Basically, as we find new virus contigs, usually they represent novel VOTUs and not VOTUs we had already seen before. So we have still a way to go here. Okay, next two things we do is taxonomic assignation and host prediction. So taxonomic assignation is done, unfortunately, again, using somewhat of a custom framework. We use shared gene with references when they are, when our contigs are very closely related to a known isolate, or we use marker genes, HMMs, this time from the VOG DB. The bottom line is like we can't really do a proper, very detailed and refined taxonomic assignation at this scale yet. I don't think we have the tool. For instance, we have not tried to do like the whole of vContact2 with the whole of IMGVR because I don't think the results will make a lot of sense. I'm happy to talk about this later, basically, but at this point, I don't think we are ready for it. If you are curious about what's in IMGVR v3, and again, it's all based on marker gene, basically this big blue bar here. I didn't go further into the taxonomy, but I can tell you these are head-tail phages, basically. These are codovirials, the one you would expect. What I'm really curious about is this part here, which is these sequences are high quality and they have no marker gene at all that can link them to even some class or even like some phylum. So I'm really curious about this one. This may be some really cool new viruses. Host prediction is a bit of the same thing. We don't yet have a super fully fleshed out pipeline that just does everything and is great. So we do, like most people, kind of bits and pieces here and there. We use BLAST to host genome. We, of course, use CRISPR spacers to do host prediction. And then we also, expand the prediction at the VOTU level. And especially when this VOTU includes some prophage, which can then be linked to a host. We have kind of benchmarked this and seen that it worked pretty well. Basically, VOTUs are consistent at the host level. Most of the time, IMGVR UVings will not come with any host information. Like the pie chart here tells you only 15% have a host prediction. It's actually not as bad as it seems because it's actually really dependent on your genome completeness. You can see here, if you just look at genome fragments, the vast majority have absolutely no host prediction. If you look at high quality or medium, what I would call medium quality is not yet official, but like 50% and more completeness, we are more in this 30, 35% of host predicted, which is a good sign, right? Like we are, if you can get a big enough piece of genome, you actually have a much higher chance of being able to link this virus to a potential host. That's good. Of course, I won't go into detail about the host breakdown because it's like such a, 10,000 mile view, but as you would expect, the hosts predicted are basically the major bacteria you would expect to find in a metagenome, Bacteroides, Firmicutes, Proteobacteria, etc., etc. So really, to wrap up what we do in terms of like data and features in IMGVR, we realize that 2.3 million sequences is a lot, and none of them are equally useful. So we want to provide more information than just this is likely a virus config. And so what we do provide is quality, taxonomy, and host that you can go in and slice and dice this database and find really what you want and the subset that is really useful for you, and not only useful, but useful and manageable. Okay, a few minutes on the interface just to wrap things up. So IMGVR is available at this URL on top right here. So it's basically IMG/VR. When you come into this page, you get this, login page, some stats on the left. But the two things that are really like the two menus that are important for you if you really want to dive into the database are this search and browse menus. And I will start with the browse part. So browse for us is really the idea, like you come in and you just want to explore what's in the database. And so we provide kind of et cetera, et cetera. When it's categorical variables, the way we have kind of let you explore the database is really by what we call this tree map. So these are interactive tables that represents the whole of predicted hosts with the areas of each square being proportional to the number of, in this case, predicted hosts. And you can click in and zoom in and kind of dive in. And eventually on the top, you also have kind of the number of VOTU. For instance, if you go and over this specific bacterial taxon, you will have like this 13,100 VOTU. So that's kind of lets you go through this, again, categorical variables. We have this little different interface that I like a lot, which is what we call by geolocation. Simply stated, it's an interactive map, and that tells you all of the metagenomes where we found some viruses. At first, by groups, and then as you zoom in into the map, you can actually get individual metagenomes with this little dots, the blue here, blue symbols, and then for each of these symbols, which are individual locations, you get a list of metagenomes, and the numbers here, which are a little small, I realize, but you should be able to see some numbers here. These numbers are the number of viral contigs, and each one of these is a link, right? You can go back to the metagenomes and to the metagenome page to get some information. And if you click on the number of virus contigs, you will get a direct page of all, a list of these virus contigs, and then you can explore them specifically. That's for the browse part. Now the search part is more like, okay, you know what you want. but you don't want to scroll through 2.3 million sequences to find it. So that's what we call the search by, the most, I mean, to me, the most important and the most useful is this search by attributes. So this gives you a page like this, and it's actually too big. I had to break it down into two pieces, but it's actually one single big form. And here you can use a number of different features to really subset the database. And here I did something that, to me is kind of a basic one. I want all the sequences that come from marine environments. I want the host predicted to be gamma proteobacteria here. And then in terms of taxonomy, I only want this autography viridae, and I want them to be high quality. So that's kind of a combination search of like, I want these high quality genomes coming from this ecosystem in this taxon, predicted to infect this host. And then you can just click go, and you will have a table of all of these various contigs to then export as in different formats, or explore online if you want to just like browse the actual sequence and annotation. Now, the important part that maybe we don't advertise enough, especially for this IMGVR database, which is still in development. If you would like to see some additional features, for instance, in these UVig attributes, you may want to search by a feature that we have not provided yet as a searchable part. Please feel free to use this contact us button that is at the bottom of the page always, and just reach out and tell us, oh, why can't I do this? Please let us do this, and so on and so forth. We are more than happy to just like get feedback from the user community and adjust as we go, especially again, as this database is still really much in progress and in development. Okay, final part of like this interface. I showed you how to browse and search for the database. Once you get to a UVig or to a config, what information do you get? You get this, what we call this detail page, which tells you like a little bit about like the actual sequence itself. In particular, we have this tab called neighborhood, where you actually have a map of the sequence. And the actual virus region that we detected is framed in this red dashed lines. If you look at the genes in the UVig, you actually can have a gene table with annotation, specific functional annotation. You can see here like some terminase, some protein of a non-function, a lot of hypothetical proteins. This will look like a regular phage for most people, I guess. And in the host prediction, you get actually the full table of which elements were used to predict the host taxon. So in this case, a CRISPR match, and you can actually link back to actually which host scaffold had the CRISPR spacer. And so you can really go back and dive into why exactly are we telling you that this phage likely infects this host, or this actually virus contigs that infects this host. Okay, very last kind of bonus and add-ons. On IMGVR, you have this virus spacer blast. If you come in with one sequence, this is not meant for like high throughput analysis, but if you have one or a few sequences that you would like to compare to IMGVR, you can do it through the online blast. Otherwise, we recommend you download the database and run the blast on your side. And that's really the last part. If you want to do more advanced analysis with IMGVR database, you can download the whole thing through our portal, and then you also have all the metadata. We are good. This is basically my conclusion slide. Yeah, right in time. IMGVR lets you do some virus exploration based on all this metagenomic data. We start to have standard for analysis and discovery and report, and we are trying to kind of implement them as we go into the database. A major challenge is this host linkage. Again, we are trying now to come up with this comprehensive annotation and user-friendly interface. We are not there yet. A lot of things still need to be standardized, and we need to also kind of use more and more of this kind of shared pipeline rather than everyone doing its custom approach every time. But eventually, I'm convinced we will get there. Now, the next step and missing part, I do want to highlight Antonio Camargo, a scientist in JGI who recently joined us and is working on kind of the next generation of IMGVR, which I'm convinced will be a really nice step forward, especially because we will update actually the detection approach. And unfortunately, we can't use out-of-the-box tools just because of the scale of data. So Antonio is looking at a number of different options to really make a new scalable detection approach, which will leverage all the improvements made recently by all of the tools we have discussed on Wednesday. And we also want to look into things like prophages and do a much better job at like sorting out which is really a virus versus which is like a false positive, like John was mentioning. And eventually, we also want to integrate much better with NCBI, KBase, EBI, EVBC, and whatnot. And that's the only slide I want to end with, and that's to thank everyone involved in all of this iteration of IMGVR. And as you can imagine, it's such a big team. But David Perez was really the leader of the first project. And then I want to, call out Krishna and Hannah, who kind of lead all the UI and interface and database of the website, which they do an awesome job. So thank you.
Great, thank you. Excellent talk, Simon. So we're going to take one super quick question, and I think this might be a longer answer, so we can cover the rest in the panel. But yeah, just quick response. What is the definition of high quality phage genome sequence, and what kind of criteria and or software execution need to be validated or done before we can say we have a complete phage ready for deposition to NCBI? So what's a high quality phage?
So in my mouth, a high quality phage means estimated to be more than 90% complete and in one piece, basically. Yeah. And this is all kind of laid out in this MyUVIC paper, this 2018 paper. So if you want more information about like why we thought about along these lines and how we went there, we kind of explain it somewhat in this MyUVIC paper. I would say, in my mind, a completeness estimation of more than 90% with a standard tool like CheckV or something like, or a comparable tool is enough to justify submitting this sequence to NCBI. I know NCBI will take it. Level of comfort varies from person to person, I would say. So some person would really not want to put anything in the database until they can actually see that they can, isolate this page in the lab, for instance. So it's up to you. But to me, yeah, you can go ahead and if you have a near complete genome, basically it's worth putting in NCBI in GenBank.